I'd like to thank Asim and the other organizers for giving me the opportunity to participate in this really wonderful celebration of an extraordinary life in science. It's really been, it's been a treat to see sort of the founding fathers of the field, I have to say. My lab is mainly interested in trying to decipher DNA regulatory codes governing the precise switching on and off of genes in the early Drosophila embryo. And this shows you, I think, one of the features of the fly embryo that makes it so good for just the pure pursuit of visualizing differential gene activity in development. The early embryo is a syncytium. You're looking at individual nuclei dividing here synchronously. So after about 2 hours following fertilization, the embryo is composed of a few thousand nuclei formed in a monolayer at the surface of the egg. And so it's very easy to gain access to these synchronous nuclei and to visualize gene activity. The fly embryo is now being used for a variety of computational and quantitative studies. This shows 500 different embryos, all within 30 minutes of each other in development, stained with different combinations of regulatory genes that control the segmentation of the embryo. And these studies are being done by a former postdoc of mine, John Reinitz, who's at Stony Brook. Now, a recurring theme in the formation of these highly localized patterns of gene expression is the use of crude regulatory gradients to produce sharp on-off patterns of expression. And one such gradient is shown here. So this is a side view of an early embryo, 2-hour embryo, stained with an antibody against a sequence-specific transcription factor called Dorsal, which is related to mammalian NF-kappa B. If I'm seeing a little skittish and a little nervous, I came late to the symposium because I was teaching at Cold Spring Harbor in the Gene X course. And the great man himself, Watson, came in to hear my lecture, which I was very proud of, but he immediately fell asleep. And he didn't just sleep. He was wrestling with his many demons during his heavy slumber and getting kind of noisy, and it was very distracting. So I'm keeping an eye on Professor Khurana there to make sure I can keep him awake through, I hope, a little crisper talk than the one I gave at Cold Spring Harbor. So I'm just a little gun shy. So this side view of the embryo shows the distribution of the dorsal protein. You can see the nuclei and the associated cytoplasm as a syncytium at the periphery of the egg, so it's just a surface view. And the protein is distributed in a broad nuclear gradient with peak levels present at the bottom of the embryo, the so-called ventral or belly surface, and lower nuclear levels in these more lateral and dorsal regions. The dorsal or top of the embryo will give rise to the back of the fly. Now, this dorsal nuclear gradient controls the patterning of the embryo by differentially regulating about 50 to 60 target genes in a concentration-dependent fashion. And these genes were initially identified in classical genetic screens and more recently using whole genome methods such as whole genome tiling arrays. The next slide shows a few examples of dorsal target genes. These are genes that are directly regulated by different concentrations of the nuclear gradient. This embryo is just an hour older at most compared to the one I showed you in the previous slide. So that dorsal gradient is getting right down to business to lead to these different localized patterns of expression. This is a multiplex in situ hybridization. The gene visualized in yellow is activated by high levels of the dorsal gradient. Those visualized in blue, green, and red are activated by progressively lower levels of the gradient. To understand how the crude dorsal gradient could lead to these precise thresholds in gene activity, we've done a whole genome survey of dorsal target regulatory DNAs, or dorsal target enhancers. We've identified about 100 enhancers and characterized 30 of them. And these are summarized here. Altogether, the dorsal gradient very quickly generates 6 distinct patterns of gene expression across the dorsal ventral axis of the embryo. And on the right, you see the summary of the regulatory DNA sequence, the enhancers, which produce these different patterns of expression. And you'll see the dorsal works in a highly combinatorial fashion, with a variety of different sequence-specific transcription factors to produce these diverse patterns. So, for example, the pattern seen in panel C, this fine line of gene expression, depends on an enhancer which contains dorsal binding sites and binding sites for a different sequence-specific transcriptional activator called suppressor of hairless, which mediates notch signaling. In contrast, this broader lateral stripe of expression depends on an enhancer with a slightly distinct sequence. This one contains dorsal binding sites and binding sites for a transcription factor, no binding sites for suppressor of hairless, but rather binding sites for a transcription factor which contains the so-called ETS domain. This is the pointed protein which mediates EGF signaling. Now, one feature of these enhancers that emerged from computational studies is the fact that there are fixed arrangements of binding sites, particularly for those enhancers that are responding to low levels of the dorsal gradient. So if you look at panel E, you see this nice lateral stripe of gene expression. This stripe comprises about 6 to 8 cells in width, and it's activated by intermediate and low levels of the dorsal gradient. There's about a 10 to 20-fold drop in the concentration of the dorsal gradient across the limits of this lateral stripe. But you see a very stable pattern of expression across this dynamic range. The corresponding enhancer has binding sites for dorsal and one of its partners in crime, the twist activator. But it's not enough just to put the binding sites helter-skelter anywhere in that enhancer. There's a fixed arrangement of binding sites whereby the asymmetric twist site points towards the dorsal site and they are closely linked. This fixed arrangement of binding sites, which we call grammar, is important in this case for fostering cooperative occupancy between the two proteins. And this ensures that the enhancer gives a stable output pattern of expression over a broad dynamic range of the gradient. And this is a general property of those enhancers that respond to low levels of the dorsal gradient over broad dynamic range. And this fixed arrangement of binding sites is seen for all 5 unrelated enhancers that produce this pattern of expression, and it's conserved in evolution among the 12 sequenced Drosophilids. Now, about 2 years ago, we hooked up with Rick Young at MIT, and in particular worked with one of his postdocs, Julia Zeitlinger, to do a whole genome survey of dorsal binding clusters throughout the Drosophila genome. This is using ChIP-ChIP and ChIP-seq assays. And these experiments also involve the use of antibodies against some of the other proteins that work together with dorsal to give these different combinatorial patterns. And altogether, the ChIP-ChIP assays identified about 200 dorsal binding clusters throughout the genome. We have reason to believe that at least 100 of these correspond to authentic enhancers, and we've tested nearly half of those. One surprise that emerged from those studies is that many of the dorsal target genes, particularly those that respond to low levels of the gradient, contain multiple enhancers for the same pattern of expression. So here you see 2 examples of the entire whole genome tiling array. These are the genes SOG and Brinker, which are activated in lateral regions in response to low levels of the dorsal gradient. The ChIP-ChIP assays accurately identified the known intronic enhancer, which regulates SOG expression, but these assays surprisingly identified another cluster of binding sites that was not predicted by genetic studies or any of the molecular manipulations we've been doing over the past 20 years. And this distal cluster, which is located about 25 kilobases upstream of the start site on the other side of this linked gene, nonetheless functions as an authentic SOG enhancer. So when you take this genomic DNA fragment, attach it to a LAG-Z reporter, express it in a transgenic embryo, you get lateral stripes of expression similar to those produced by the primary enhancer and seen from the endogenous gene. And we refer to these remote secondary enhancers as shadow enhancers. People ask, well, why shadow enhancers? Well, I'll tell you why. If we called them duplicate enhancers, we would have gotten a paper maybe in development. But by calling them shadow enhancers, something kind of fancy, well, we got a science paper. So that's just a little lesson to the younger people. In any event, sometimes these shadow enhancers are located within the introns of neighboring genes. There's no way you're going to find that through any kind of conventional gene fusion assays. The only way to identify these cryptic enhancers or shadow enhancers was through the ChIP-ChIP assays. Again, these assays accurately identified the known 5' enhancer that regulates Brinker, but this intronic enhancer also serves as a shadow to regulate Brinker. As I mentioned, almost all the dorsal target genes that respond to a broad dynamic range of the gradient, particularly low levels of the gradient, seem to contain shadow enhancers. And this is not just true for the dorsal gradient, but also for the more famous bicoid gradient, which controls head-tail patterning. So I showed you the dorsal gradient, which is distributed up and down in the early embryo. The bicoid gradient, studied in Nusslein-Volhart's lab for many years, is distributed anterior to posterior with peak levels present at the anterior tip, the future head, lower levels in central and posterior regions, the future tail of the embryo. And the bicoid gradient regulates a variety of target genes in a concentration-dependent fashion. These are collectively known as the gap genes. The most famous of these is shown here, the hunchback gene, which shows a very sharp border of gene expression. It's activated by high and intermediate levels of the bicoid gradient, and over maybe a twofold drop in the level of the bicoid gradient, you get this sharp posterior border in the activation profile of the hunchback gene. And this has been subject to a lot of studies, including both computational and mathematical modeling studies. All of these studies invoke one enhancer for this expression pattern. This is the classically known enhancer identified about 20 years ago in conventional gene fusion assays, which straddles this proximal hunchback promoter. But a graduate student in the lab, Mike Perry, through chip-chip assays, identified about 5 kilobases upstream a shadow enhancer, which produces a very similar pattern of expression to the classical enhancer. And if you look into that shadow enhancer, it has the similar kind of logic, 3 linked bicoid binding sites separated by helical phasing and so on and so forth, obviously engineered for optimal cooperative interactions. So why shadow enhancers, and why the preponderance of shadow enhancers with genes that are responding to gradient information? And studies done by a graduate student in the lab, Alistair Boddicker, suggests that it has to do with resilience to perturbations, either in the environment or genetic perturbation. And so here you see the expression of 2 genes, SOG and NU3, in a very mildly mutant embryo, a dorsal heterozygote, 1 copy of dorsal rather than 2. And this scales, so the dorsal gradient's composed of half the concentration of the protein than wild-type embryos. Now, in this very mild mutant, these, for the most part, will develop fine, particularly at low temperatures. The SOG expression pattern is activated just fine in this dorsal heterozygote. But NU3, which should show a very similar pattern of expression, and it does in normal embryos, shows an erratic activation profile in this heterozygote. SOG has a shadow enhancer, as I showed you. NU3 does not. So it looks like the shadow enhancer helps ensure that you get proper activation in gene expression even upon perturbation, either genetic, and we've also done similar experiments by raising the temperatures to the very threshold of what the fly embryo can tolerate and develop in. The shadow enhancers help ensure a reliable response. And I don't think there's anything fancy about it. If you have 2 enhancers rather than 1 for the same pattern, then you're more likely to get critical activation of the gene at the right place at the right time, even when there's perturbation. Now I'm just going to finish up by talking about the second major surprise. So first of the shadow enhancers. Another surprise that emerged from the chip-chip and the chip-seq assays done collaboratively with Julia and Rick is the finding that RNA polymerase II, POL2, is already present in the promoter regions of most, not all, but most dorsal target genes before they're activated in the early embryo. And an example of this is summarized here. So this shows a gene you've seen previously, SOG. SOG is regulated by this intronic enhancer and this remote shadow enhancer. And Julia did comprehensive chip-chip and chip-seq assays with antibodies against RNA polymerase II. And in the early embryo, prior to the time when SOG normally comes on, you see this nice spike of polymerase binding right at the 5' promoter region. So why stalled polymerase? About 10% of the genes in the Drosophila genome show this kind of polymerase stalling. Almost all of the critical developmental control genes that you think about have the stalled polymerase. So this would be the Hox genes, genes that encode notch, winglets, hedgehog, FGF signaling, and so forth. Almost all of those genes have stalled polymerase. Now, one hint comes from pioneering studies done by John Liss over the past 20 years, who's shown in the case of the heat shock genes, the stalled or palled polymerase greatly accelerates the response of these genes to heat shock, to stress. That makes sense. I mean, you don't want to fumble around and take forever to mount an SOS response. So having the polymerase there gives a rapid response. Comparable promoters lacking the stalled polymerase take much longer for their induction by heat shock. And it's possible to think about developmental control genes in the same way as stress genes. They have the stalled polymerase. They are repressed but poised. They're ready to go. When the right signal comes along, like a transient sonic hedgehog set of cells, then the gene is triggered and it's off to the races. And I think that's right. So I think the stalling does help give sharp on-off switches in gene activity in response to transient signals. But a graduate student in the lab, a biophysics student in the lab, Alistair Boddicker, has obtained evidence for a second mechanism for the stalled polymerase, which I'm going to now finish up with. And the thinking goes like this. Here's a promoter, and this is a gene that's regulated through classical recruitment. I mean, I was taught, most of the textbooks say, that to turn a gene on in development, activators like Dorsal bind to the enhancers and somehow ultimately recruit RNA polymerase II to the core promoter. Now, problem with this is that the binding of polymerase is a bit messy, and it has a high failure rate. So oftentimes, polymerase will bind, but it fails to become phosphorylated. It fails to undergo promoter clearance and melt the DNA, and so it falls off. Now, if you think about it, that means that a field of cells, all with a gene that's going to be induced by the same inducing agent, should come on in a stochastic fashion. And this variability, or this noisy induction from cell to cell, reflects the high failure rate of polymerase to bind to the promoter and successfully undergo promoter clearance. In contrast, genes like the developmental control genes that I've been talking about that have the stalled polymerase should come on in a much more synchronous fashion. The polymerase is there. It has already cleared the checkpoint. In other words, it's already bound. It's already undergone promoter escape. And it's sitting there awaiting instructions. And we know in some cases that instruction is a protein called PTEF-B. If we recruit that to this promoter, PTEF-B phosphorylates the C-terminal tail of pol II, and that's sufficient to release, at least not in all of the stalled promoters, for about half the stalled promoters, PTEF-B is sufficient for the release of the polymerase. So in principle, it's a much simpler process. It's one step, boom, and it's much cleaner, and it probably has a much higher success rate because the polymerase has already cleared the checkpoint. So to test this idea of stochastic noise associated with promoters that require recruitment versus nice synchronous patterns or coordinate or uniform patterns of activation in a field of cells when you look at genes with the stalled polymerase, Alistair Boddicker developed a highly quantitative in situ hybridization method where he's detecting the first nascent transcript made, cell to cell, upon the activation of the gene. And he's using computational methods to look at this quantitatively. And this just shows an example of his analysis. This is now comparing the moment of activation of two genes, SOG on the right, THSB on the left. Both these genes have very similar kinds of enhancers. They're both regulated by the same low levels of the dorsal gradient, but they differ in one important way, and that is that THSB lacks stalled polymerase, whereas SOG has this nice peak of polymerase, stalled polymerase, at the core promoter. And at the moment of induction, you can see that THSB shows a salt and pepper pattern of activation through the field of cells where the gene is expressed, whereas SOG shows a fairly uniform pattern of expression. And because Alistair has looked at so many embryos, he can put a time on it. There is only a 2 to 3 minute lag between the appearance of the first nascent transcript in one of these cells in the field and the expression of nascent transcripts in all of the cells, just 2 to 3 minutes. In contrast, in the case of those genes that lack the stalled polymerase, there's a 20 to 30 minute lag between the first appearance of the first nascent transcript in one cell and then the appearance of nascent transcripts in all of the cells. So this is synchronous versus stochastic induction. Here he's done a double labeling, and this is just about, it's one minus the penultimate slide. I'm there, baby. I know it's late. I know you guys want to go. So this is just looking down on the belly of the embryo. This embryo was double labeled with probes that detect the initial nascent transcripts of two different genes, MES2 and MES4. MES2 has a stalled polymerase, MES4 does not. You can't see much, but here's the high magnification view, and if you look carefully, you'll see that all the cells show green dots. Okay, that's the stalled gene, so it's showing the uniform activation, but only about half the nuclei show the red dots, and it'll take another 20 to 30 minutes for all the cells to start expressing the red dots. That 20 to 30 minutes may not sound like a lot to you, but from my perspective, that can introduce a lot of developmental variability, given the speed with which this fly embryo is forming. I mean, basically, you go from naive, totipotent embryonic nuclei to absolutely committed nuclei that will follow specific pathways of differentiation in just 45 minutes. All right, so 20 to 30 minutes is, in my opinion, a lot of potential noise and uncertainty. And so this just shows, statistically speaking, Alistair sees two classes of genes with respect to their initial activation patterns. Genes, you can tell from the light coloring, which show a lot of variation from nucleus to nucleus at their moment of activation, and genes that show nice uniform synchronous patterns of expression. These 5 genes, which show a lot of noise, all lack stalled polymerase. These 9 genes, which show fairly uniform activation, all contain stalled polymerase. So there's a very close link between stalled polymerase and synchronous activation. Why synchrony? I think it's a real property, a real manifestation of the metazoan embryo. Now, yeast essentially lacks stalled polymerase. And if you think about yeast, when they're challenged by changes in carbon source, osmotic shock, you want a stochastic response. That's part of its adaptation. You want to make sure that a few of those cells will give an appropriate response where they'll survive. It's just the opposite when you're talking about coordinate gene control in a metazoan embryo, where you have fields of cells that have to function as one, that are brought under very stringent coordinate control. So I think the transcriptional synchrony is an important mechanism of coordinate gene control and development. It ensures the orderly deployment of the complex gene networks that control embryogenesis. And I just want to thank the people who did the work. I discussed 3 mechanisms of transcriptional precision in the fly embryo. Grammar, where you have fixed arrangements of binding sites in the target enhancers. This was done by Dmitry Papensenko and Rob Zinson. Shadow enhancers, which provide resilience in gene expression to genetic and environmental perturbations. This was done by Jungwoo Hong and Dave Hendricks. The stalled polymerase was discovered by Julia Zeitlinger while she was a postdoc in Rick Young's lab, and I showed you evidence that the stalled polymerase is important for coordinate activation for transcriptional synchrony in the early fly embryo. And I'll stop there.